Hello, everybody. And before I start, I would like to thank Montserrat Samson and the organizer of this meeting for giving me the opportunity to present the progress of our work. Today, I will talk about calcium-entry units, new intracellular junctions recently characterized by our laboratory. The sarcoplasmic reticulum of skeletal muscle fibers is composed of two main domains. Those regions that are closely associated to the tubules, marked in yellow, which are named the terminal cistern or junctional SR. These regions are darker because they contain calsequestrin, a protein that accumulates calcium in proximity of the sites of calcium release, the ion receptors. And then there are these other regions named free SR or longitudinal SR, which may be located at the A band or at the I band of the sarcomere. During my presentation, I will show you that the I-band SR is capable of significant remodeling during exercise. Now, free SR does not contain calsequestrin under normal conditions, but is rich in Zerka pumps, which operate calcium reuptake. Calcium handling in muscle, though, is not just under the control of EC coupling, but is the result of the cooperation on different mechanisms. Briefly, following the depolarization of the transfer tubules, the interaction between the ryanodine receptors and the dialpropyridine receptors activates calcium release and contraction of myofibrilles. Now, the majority of this calcium is then quickly pumped back by Zerka pumps to refill the SR with the consequent relaxation of the fiber. And then another little amount of calcium also enters mitochondria through the mitochondrial calcium uniporter. How much? Well, definitely enough to activate the aerobic ATP production. And then another little amount is pumped outside the fiber through the plasma membrane calcium pumps. However, when muscle is stimulated repetitively, like during a prolonged exercise, the total amount of calcium within the SR decreases to a level that contributes to muscle fatigue. And the accretion of the SR is known to activate a ubiquitous calcium influx mechanism called store-operated calcium entry or SOCI. Two proteins are involved in this mechanism. SIM1, a calcium sensor in the lumen of SR, which interacts and activates ORI1, a crack channel of external membranes, so either plasma membrane or transfer tubules. And calcium finally enters from the extracellular space to replenish the SR. The skeletal muscle SOCI has been first reported by the group of Ogawa in the 2001, and then by a work of Rios and colleagues in the 2003. Now we also know that abnormal SOCI contributes to muscle dysfunction in different physiopathological conditions, that mutation in SIM1 and ORI1 are linked to diseases such as tubular aggregate myopathy, and that SOCI in muscle is important to limit muscle fatigue. Now, for a few years, it has been proposed that SOCI must occur at the same junctions mediating EC coupling, the triad. However, this hypothesis never fully convinced us, and for these reasons, we decided to start looking for the sites of SOCI in skeletal muscle fibers. And keeping in mind that SOCI play an important role in limiting muscle fatigue, we simply designed an incremental running protocol to fatigue adult mice. The entire protocol was about 1 hour long, then animals were sacrificed, muscle processed. And when I look in electron microscopy at the EDL muscle from mice pre-exercise on the treadmill, well, my excitement was up to the stars. I still remember those days sharing my enthusiasm with Feliciano Protasi and Antonio Michelucci, the postdoc that was performing the fatiguing protocol. So after the running protocol, the sarcotubular system at the I band between triads and Z lines was remodeled to form these peculiar stacks of flat and parallel cisterne that we call as stacks. You see that in control sample, usually VSR at the I band is very convoluted, many vesicles. Actually, the quantitative analysis that is not shown here demonstrated that some of these stacks are also present in control muscle, but they are smaller, formed by fewer elements, and not as frequent as in exercise mice. We went on with the experiments, and using the ferrocyanides, which form an electron-dense and dark precipitate into the tubal lumen, we demonstrated that the exercise induced not only the remodeling of ESR members, but also the elongation of the transverse tubules. So the tubules elongate into the bend toward the Z-line to make contact with the ESR stacks. And the continuity of a triadic tubule with one of this newly formed structure is very well visible in panel C after false coloring the two membrane compartments in yellow and green. And finally, using the confocal microscopy, we also show that this remodeling of membranes at the eye bend result in an increased colocalization of ORI1 with SIM1, the two protein of SOCI. After exercise, the red-green, so SIM1, and the red staining, SIM1, and the green staining, ORI1, were often overlapped, resulting in this strong yellow signal. So based on this finding, we propose a model summarizing changes occurring during the exercise. The remodeling involves elongation of the transverse tubules into the band toward the Z-line, fusion of the vesicle into larger elements, and flattening in ESR cisterna with their final docking to with tubules to promote the assembly of new intracellular junctions that we called calcium entry units, as they contain SIM1 here in red and ORI1 here in green. These junctions may provide a preferential site for calcium entry during repetitive muscle activity. And when we were working on this data in the 2015, a paper was published by the laboratory of Clara Franzini Armstrong, post-author Stefano Perni, showing the appearance of new structure in X cells following SIM1 and ORI1 overexpression. And the similarity with our junctions in EDL exercise mice was quite obvious at our eyes. So I thought, to functionally test our hypothesis, we subjected intact EDL muscle dissected from control and from mice pre-exercise on the treadmill to an ex vivo stimulation protocol. As you can see in panel A and B, in presence of extracellular calcium, the muscle from pre-exercise mice are more resistant to fatigue, the curve in black, than muscle from control animals. We repeated the same experiments in zero extracellular calcium or in presence of calcium but in a solution supplemented with two SOCI inhibitors, VTP2 and 2-APP. The results of these experiments indicates that when calcium entry is prevented, the drop in force in control muscle is smaller than the drop in force in EDL muscle from pre-exercise mice, indicated that indeed these muscles are more dependent from extracellular calcium. So after having published the first paper in Scientific Reports in 2017, in collaboration with the laboratory of Robert Dixon and Antonio Michelucci, we also did two other sets of experiments. We performed the manganese quenching, a technique considered the gold standard to measure entry of divalent cations from the extracellular space. And briefly, in these experiments, single dissociated FTD fibers were first incubated in a calcium-free solution containing Furia-2 and supplemented with SERCA inhibitors in order to fully deplete DSR and maximize the activity of SOC. And then in this experiment, extracellular calcium is substituted with manganese, and when manganese enters, the fibers results in a quench of Furia-2 fluorescence. Here, you see that in pre-exercise mice, manganese quench, the trace in blue, is more pronounced than in control animal. And this effect was not present in different mouse models that are knockout for ORI1, meaning that indeed the entry of manganese into the fiber occur from ORI1 channels. In the second set of experiments, we determined the time course of disassembly of calcium entry units. So we hypothesized that calcium entry units which assemble dynamically during exercise would disassemble following recovery. And the results of these experiments are summarized in this slide. We determined that the plasticity of T-tubules is much greater than that of ESR. Indeed, as it is modeled in the cartoon, the time course of retraction of T-tubules from the eye band is faster, less than 6 hours, than the time that takes the ESR to disassemble, more than 12 hours. The time course of T-tubule retractions creates a nice parallel with the rate of manganese quench, visible if you compare these two bar graphs here. So we concluded that we may say that the key event in the assembly of fully functional calcium entry units is indeed the elongation of the transverse tubules, which contains ORI1 from the triad. The tubules elongate into the band to associate with ESR stacks, which contains SIM1 proteins. Okay, what am I doing now? I'm working in two main different directions. I'm investigating soaking calcium entry units in muscle during postnatal maturation. In the next two slides, I will show you preliminary data in mouse fibers at 2 weeks of age. Actually, in this project, we will check also different stages of muscle maturation and differentiation, such as myotosis. And second, we are also investigating whether calcium entry units also assemble in slow twitch fibers of soleus muscle, and if their morphology is similar to those found in EDL. As you can see here, for this second task, we employ two mouse models lacking calsequestrin, a calsequestrin 1 null and a double calsequestrin null mice. Why? First, because we could not get a definite answer in exercise wild type mice, possibly because we could not fatigue sufficiently soleus in vivo. And also, in addition, we know from a paper published in the 2010 that the lack of calsequestrin causes severe ESR depletion during repetitive stimulation, and that the EDL fibers from these mice containing pre-assembled calcium entry units that enhance both constitutive and SOR-dependent calcium implants. This data have been recently published in the 2020. So let me start by showing to you the data collected at 2 weeks of age, mice. The experiments performed by Antonio Michelucci in the laboratory of Robert Dixon. What you see here in panel A, these are representative traces from manganese quenching of Cura2 fluorescence experiments performed in single FTP fibers, enzymatically dissociated from 2 weeks and 4 months of age mice. These data show a more strong rate of manganese quench in 2 weeks old mice than at 4 months. If we calculate the steepness of these traces, that is the maximum rate of manganese quench at 2 weeks of age is about 4 times larger than that at 4 months. So strongly indicated that the stocking mechanism is upregulated at 2 weeks. How can we explain this? Well, our previous work indicates a correlation between the use of external calcium and the presence of calcium entry units. So we decided to look at calcium entry units in electron microscopy. In panel A, you see that at 4 months of age, most tubules pointed by black arrows and stayed in black with ferrocyanides are almost exclusively transversally oriented and constitute the central element of the triads, though positioned at the A-I band transition of the sarcomere. On the other hand, at 2 weeks of age, the sarcotubular system is not ordered as in adult fibers, and many tubules are still longitudinal. And this actually was already known thanks to the work of Clara Francini Armstrong, because this coupling system undergoes during a postnatal maturation structural modification involving a progressive transition from mostly longitudinal to almost exclusively transversal. What is new is that if we look closer to the longitudinal tubule at the I band, we now see that this tubule make constitutively assembled junctions with the ZAR stars. This is very well visible in these images and also in cross section, where I labeled, I false labeled the yellow PSR, and tubule is this one in black. We perform a quantitative analysis of the 2 different components of calcium entry units, so it's a stacks and tubules, and all parameters are 3 to 4 times greater at 2 weeks compared to 4 months. This data indicates that calcium entry units are constitutively assembled in young fibers. We perform a similar electron microscopy analysis also in adult fibers from Soleo. And for the reason I anticipated in the previous slides, we stain with ferrocyanide not only soleus from the wild type, but also from calsequestrin 1 null and double calsequestrin null mice. As it is visible, if you compare these images, the wild type with the knockouts, we found an abundant presence of longitudinal T-tubule at the I bands, in fibers from knockout animals. And this is similar actually to what we already described in calsequestrin 1 null mice for EDL. The only difference in the appearance of calcium entry units is that in Soleo, T-tubules usually makes contact with just one SR cistern. Rarely we found stacks formed by 2 or multiple SR elements like in EDLs. We quantify the elements that form functional calcium entry units, number of stacks, the extension of a transverse tubule, and number of SR T-tubule junctions, oral significant more abundant in fibers of calsequestrin knockout animals. Actually, in double knockouts, more than in calsequestrin 1 null. Just one note, this is a longitudinal triad, and in which I label here red the RNA receptors, and again in yellow you see the sarcoplasmic reticulum. Well, longitudinal triads are obviously excluded by the quantification. To confirm that the presence of pre-assembled calcium entry units at the I band enhances SOPI, we again performed the manganese quenching experiments. The experiments were carried out following SR depletion, these 3 the panels up here, and without depletion, 3 panels on the down here. Following the store depletion, fibers from calsequestrin 1 null and double calsequestrin null mice display a significant increase of quench after application of the manganese, extracellular manganese. And the quantitative analysis revealed indeed that the maximum rate of quench in fibers from both calsequestrin 1 from both mouse models is about 2 times greater than in the wild type, a result even supported by the frequency distribution analysis showing a clear shift toward the larger rate of quench. In the absence of pharmacological store depletion, while in wild type fiber sequence was completely abolished, a significant rate of manganese entry was still present in both genotypes, strongly suggesting that the presence of a constitutively active influx mechanism in these mouse models. We finally verify the expression levels of C1 and ORA1 in soleus by performing a Western blot analysis. The results obtained from these experiments demonstrated that in soleus muscle, while the relative expression of both long and short C1 splice variants were not significantly different between the three groups of samples, we observe an increment of about 2-3 folds of ORI1 in homogenids from cat sequestering 1 null and double cat sequestering null mice, respectively, compared to wild type. In this slide, I summarize the findings of the most recent experiments. I show to you that at 2 weeks of age, calcium entry units are constitutively assembled, possibly due to a greater amount of longitudinal titubule. I show you that calcium entry units are also present in soleus muscle, but they are usually formed by a single SR element associated to the titubule. From all the results I presented to you, we can conclude that the presence of calcium entry units always correlates with an enhanced contractility and entry of divalent cations from extracellular space via SOCI. Before to conclude, I would like to thank the people that contributed to this data. Giorgia Rastelli, a postdoc in my laboratory, Feliciano Protasi, Laura Pietrangelo, Antonio Michelucci, that is now getting his position at the University of Perugia, and Robert Dixon, and all of you for your attention. Thank you.